Okay, in this video we're going to talk about how genetic libraries are screened. So let's talk just to recap what a genetic library is. Right, there's two kinds of genetic libraries here. Right, we have got a cDNA library. Okay, so this is cDNA, and this is we've got a Petri dish there, and on the Petri dish we have these are colonies of E. coli. Okay, each one of them is a separate colony of E. coli, and the whole library there may be several hundred or thousands of those. I've only drawn a few there, and each of those colonies all contain the identical bacteria. They all come from one bacteria, and in the bacteria you have those red circles, and they are plasmids. There's bits of circular DNA, and on a larger scale here from this plasmid, the red DNA is the plasmid DNA, and the yellow DNA is a bit of DNA that's been inserted into there. In the case of the cDNA library, cDNA stands for copy DNA. All the mRNA from the cell has been extracted, and using the mRNA as a template, you have made multiple bits of copy DNA and inserted them into the fragments. So inserted those fragments into plasmids, and the plasmid we call the vector, the carrier of that DNA. So we want to find out, when we're screening that colony, screening that library, what bit of DNA is in which colony. And you definitely wouldn't be interested in looking at every single colony. You're probably looking for a certain specific gene. Let's say, for example, we're looking for the alpha chain of hemoglobin, as an example. And these kind of experiments were carried out in the early 1980s on the alpha chain. I'm just using that as an example. Lots of other proteins were investigated in the same way. Okay, now here we have got a genomic library. So the genomic library is different. It's an entire human genome, or whatever animal we're looking at, and it's being chopped up into smallish fragments, maybe 20 kilobase pairs long, and those fragments have been incorporated not into plasmids because they're a bit too big, into these things which are called bacteriophages. One called bacteriophage lambda, which infects E. coli, and those bacteriophages, like viruses, which infect bacterial cells, they kill the cell, and the cell ends up making multiple copies of the virus with its DNA in there. Now, so here we've got green is a lawn of E. coli, and each of those yellow spots really is like a translucent spot, is a colony of phages, of bacteriophages. So that's all the E. coli there have been lysed, they've been killed, and they've just, those E. coli have been used to manufacture lots and lots of those colonies. Okay, so in each bacteriophage, you've got the red DNA, which is the host DNA, the bacteriophage DNA, and the yellow bit is the human DNA, which has been inserted in there. Okay, now we need to find out what's in each of those colonies. You probably have hundreds of thousands of those colonies in an entire library. So rather than one plate, you probably need like 20 Petri dishes with like 10,000 tiny colonies on each one. Right, so anyway, the procedure for looking at the DNA is basically the same for whether it's a cDNA library or a genomic library. So what happens is the Petri dish, there's a Petri dish, placed with the colonies on it. Placed over the Petri dish is this blue thing, which is a nitrocellulose filter. And the DNA absorbs onto the surface of the nitrocellulose. You leave it there. So you take that off, and you have now got a copy of the Petri dish with DNA, bits of DNA, and of course they will be invisible because it's too small, you won't be able to see them. The next thing to do is to then, well, you denature the DNA. So you denature. What that means is you make it into single-stranded. You make it into single-stranded DNA, and you do that usually by adding sodium hydroxide, strong alkali to it, and it will split it up. And what that means then is you'll have a single-stranded DNA, and on the surface of the DNA, on the surface of the, so I'll draw it like this, there's your bit of plasmid, bit of virus, bacteriophage DNA, and here in it we have our bit of human DNA that we're actually interested in. And all the little bases are going to be sticking up they're going to be fairly accessible to form hydrogen bonds. So let's say A, T, G, C. I'll just do 5 there. Okay. So what you need to identify the say looking at the alpha gene, we need to get a partial sequence of that. You don't need the whole sequence, you just need about 20 of the alpha globin gene. You need about 20 nucleotides long. Now how you get that is there's various different ways in which you can get it. But one way, and the most obvious way, is a little bit of the protein is sequenced, find out the sequence of amino acids and work out backwards what would be the nucleotide sequence for a protein with those amino acids. So you make an oligonucleotide, I'll do that in purple. Which is complementary to that sequence. And you make that radioactive by nucleotides are made up of course, they're made up of sugar, base, and phosphate. And instead of using normal phosphorus, you use 32. Normal phosphorus is 31. Use 32p phosphorus, which is radioactive. It's a beta emitter. More recent methods, instead of using radioactive phosphate, you can use a fluorescent tag on the nucleotide, and that's safer for the people who are working with it. But the original way was to use 32 phosphate, which is radioactive. So this whole thing is radioactive, and you place the mixture of your, it's called a probe. Okay, this is now called a probe, and you place that onto the nitrocellulose filter. And what will happen is, so here's little bits of probe, they will not stick to any bit of DNA unless it's got this, unless it's complementary to the sequence. Say we're looking for that sequence, it'd be too short to look for this, is only 5 long, but let's just imagine it's 20. So our probe would be T, A, C, G, G. Okay, so that would hybridize. It would form, sometimes it's called anneal, sometimes hybridize. But what it means, it forms H bonds to the complementary strand. Okay, so we'll only stick to the complementary strand. So we're going to have from one of those little ones, I just draw it there, say that one. Our oligonucleotide probe has hybridized to the DNA in that particular colony, and none of the others because they don't contain the alpha globin gene. You wash it to get rid of all the unbound probe, and then you get your nitrocellulose filter and you place it over, you got the filter, and on top of that you're going to place a piece of photographic film, And what that will do is your photographic film afterwards, when you develop it, you'll get maybe one spot, and that shows you that this spot is the one that contains the, in this case, the alpha globin gene. That's what we're looking at. So then you go back to your original library. Let's say it's the cDNA library. So here is our Petri dish. You locate the spot. So you've got loads of spots there. And you decide, well, that one contains the cDNA for the alpha globin gene. So then you can take some of this, and you can grow this bacteria. And you're going to get lots and lots of copies of your plasmid. So here's the plasmid with a bit of DNA. That will be the cDNA for the alpha globin gene. You can grow lots and lots of that, and you can get as much of this cDNA as you want. You can carry on, literally as much as you want. You just grow more and more bacteria. And then you probably want to chop it out of the plasmid, because there's a restriction enzyme you put it in with, the green cut there, and that will chop that bit of DNA out, and then you can purify that on a basis of size from the plasmid DNA, and you've got lots and lots of this cDNA for the alpha globin gene. And then you could do what was done in the early days. You could sequence the entire nucleotide for the cDNA. Now the cDNA library is usually easier to investigate first, because it's smaller fragments. You don't have all the mRNA. You've cut out the introns, the non-coding regions, and so forth. But this makes a useful probe itself now. This cDNA, you can use that to probe the genomic library. So here's our genomic library. Got all our little spots on there. You can use this bit of cDNA to make, and you can label it with 32p nucleotides, and you can so you can use it. That cDNA is used as a probe. So that will hybridise to any of those spots which contain bits of genomic DNA which code for the alpha globin gene. Do exactly the same thing again, nitrocellulose filter, incubate it with the radio labeled probe, which you've got from the cDNA. And then you might find, you've got 3 colonies out of several, 20, 30, 40, 50,000, you might have 3 colonies, which contain fragments for the alpha globin gene. That can then be sequenced. You can knit those 3 fragments together, and you can work out the sequence of the whole sequence of the gene. So I'm drawing these as, these are coding regions, so they are called the exons. And these red bits, these are the introns. And then you can also find out from the genomic library, in green, these are called the flanking regions. And very often the flanking regions, they contain things like promoters, if you've done promoters, which are sequences to which other proteins bind and make that piece of DNA be translated into mRNA. So you can work out how the gene, not only the sequence of the gene and the intron-extron structure, but maybe work out something on how the expression of that gene is controlled. So I've used the alpha globin as an example here, but many proteins were investigated in this way. Many genes for many proteins investigated this way. Prior to the human genome sequencing, which started about late 87 or 88 or something, before then, there'd been a good 10 years where people had been sequencing the nucleotide sequence for a lot of genes, human genes and genes from animals, using this method.